in chapter 24, we are going to talk about the fungus singular or fungi as in plural. So, this is the first group of organism that we are going to discuss, which are eukaryotic. That means that they do have membrane bound organelles in the cell. Now, we did not go into the detail of the cellular biology of the eukaryotic cell because that is presented in an entirely different course, which is the biology 8420 cell biology. That's so in contrast to the bacterial cell that we have discussed the cellular morphology in the detail. We did not talk about much detail about the fungal cellular morphology, which is very similar to any eukaryotic cell. But there are some modifications. So here in this chapter, we are going to look at the very basic idea about the fungi and how it is encompassed in microbiology. We are going to see the classification, and then we are going to discuss some of the general characteristic of them, their reproduction, different types of reproduction that we definitely see in these groups, as well as we are going to discuss some of the important the fungi as per their recent classifications. So the first thing, the major fungal groups, there are many different ways that historically the fungal groups are actually placed. But this is basically the latest group, which is essentially evolutionarily defined, the phylogenetic by phylogenetic analysis. This is how the fungus groups are evolutionarily placed, and this is one of the most recent classification. So the first that we are going to talk about is the zoosporic fungi. And this is zoosporic fungi, as you can see that the zoo, that means animal, as at one point of time, they were considered as protists. That means that they are animals, single cellular protozoan animals, and many of them actually do have some of these flagella in parts of their life cycles, and they were considered as protists. But now we know that these are from their sequences of the and evolutionary significances. We know that these are the genuinely true fungi. And there are two groups. The one is known as these microsporidia, and this microsporidia, these are obligate intracellular parasite, and the picture that you are seeing right here, it is known as Idhasridia. So you can always access the alternative text. The detail is in there as well as in your textbook. This is Idhasridia, and it actually is live as an endosymbiont with the Aedes aegypti mosquitoes. And exactly what is the role of these microsporidian symbiotic fungus is not well known. It is considered that it is the part of the microbiome of the mosquitoes. Whether there are many different research which is going on based on whether they have any developmental characteristics, or whether there are any of the reproductive advantage that they have, or in terms of disease spread, whether they are advantageous to have these between microsporidian Idhasridia infected mosquitoes or not. Those are being studied. Another group of the zoosporic fungi is the Chytridiomycota. And the word Chytridio starts from this word that chitin. Chitin is one of the we are going to talk about that. It is one of the polymers that is present in the cell wall of the fungus, and based on that, it is known as a Chytridiomycota. And this is the picture is an allomyces. These are found in the mud. So this is present all over the place. So this is one of the Chytridiomycota, and we're going to talk about some of the other species as well. The next one is the zygomycetious fungus. So sometimes we call this as a zygomycetes, and the zygomycetus is essentially the adjective. So zygomycetes is the noun, and the zygomycetes fungi is the adjective of that. So within the zygomycetes fungi, we have two groups. One is the mucormycota, and the picture that is in here is the pilobolus, and this grows upon the animal excreta or the dung. And you can see that these are the structures which are called the hyphae, and these are non-septate hyphae. And we're going to talk about and define all of these. And at the tip that you can see that these are the spore-forming structures, and we're going to define those structures as well. On the other hand, this other group is the glomeromycotina, and the glomeromycota is essentially all the mycorrhizae in the plants are with this. So the vascular plants do have these fungus in close association with their root system, and it is a symbiotic relationship, and they help them in nutrient recycling, the nitrogen fixation, and many different activities. So essentially, these mycorrhizae, which are present in shrubs, subtropical, tropical plants, in vascular plants, these are essentially very essential for the normal survival of many of these plants. So this is mycorrhizae is one of those common things. And here you can see that these are the plant roots, and these mycorrhizae are essentially the endosymbionts, which essentially stay with these vascular plants. So these belongs to this glomeromycota. After that, we are going to looking at the group which is called the dikarya. And karyon means nucleus, and dikarya essentially is the two nucleus. So what does two nucleus means? So all these fungi, they do have asexual reproduction, whereas the fish, the nucleus is divided into two by mitosis, and so on, and the cell divides into two, and that is the regular sexual reproduction. But we have seen there is a sexual reproduction, and in sexual reproduction, the two compatible fungi, two individual fungi of the same group come together, and they fuse, and the two nucleus come together. But what happens that there is a stage which is called a dikarya stage, where the two individual nucleus remain separated for some time. So there is fusion of the cytoplasm. So the cells actually fuse, the cytoplasm is actually fused, but there is two distinct nucleus. Each one of them contain haploid, or a single copy of in the entire DNA. Now, what we see that after some time, at a certain stage, that these nucleus actually fuse together to become diploid or 2N. And after that, again, there is a meiosis that happens. That means a reduction division, from where the haploid spores are formed and they are spread in this process. So that's basically the name behind the dikarya. And the ascomycota, or the ascomycetes, sometimes known as ascomycetes, these are the cup-like structures that you are seeing, that there are many different ascomycota. They form these ascospores. We are going to take a look at those. And this is right here is Coquina tricoloma, and this is found in Costa Rica, and the ascospores, they formed inside the cup from where they are dispersed. So the next one is the basidiomycota, or basidiomycetes. These are all the mushrooms that we usually see, and most of them are macroscopic. Some can be a very large. Even some of these mushrooms can be almost like 1.5 and 2 feet in diameter. And what you are seeing right here, this is a oyster mushroom. The name of this is Pleurotus stratus. So this is an overview of the 6 major groups of the fungus. Now let's take a look at some of these terminologies. So the mycology is the study of the fungi, and the mycologists who actually study the fungus. And the mycosis is a disease caused by the fungi. Many of these diseases affect the human beings as well as the plants. The animals all have these mycosis or fungal diseases. And the mycotoxicology is the study of the fungal toxin and their effects. Some of these fungal toxins are extremely potent and dangerous, and many of them are actually lethal. So these are some of the overview of the of the terminologies. And another important thing is that when we essentially have a generalized name, mycota, these means a fungus. Sometimes we could call this as instead of mycota, which comes from the mycology. We also use another term, which is a mycetes. And mycetes originates from the word that many of these funguses have a mycelium. And these are essentially, we're going to talk about that in detail. They have these thread-like structure, which is known as the mycelium. That's a general morphology of many of the funguses. So sometimes we call them as the mycetes as well. So mycota, mycetes, these are two general terms that are used to describe many of these fungal group and interchangeably used. So after that, we are going into a little bit more detail about the fungal groups, and let's take a look at these. So the first, we are looking at the zoosporic fungi, which is a microsporidia. These are almost all obligate intracellular parasite. That means that they have to be inside a cell to survive, and usually these are animals. They do not have a free-living form, and they lack mitochondria, peroxisomes, kinetosomes, cilia, centrioles, and sometimes they do have an organ called the mitosomes. And presence of these actually, essentially, initially, it is thought that they are protists, but they are not. The spores also have this inner chitin wall, and chitin is a polymer of N-acetylglucosamine. So N-acetylglucosamine bonded to another, and a polymer of N-acetylglucosamine makes the chitin. And there is also the outer wall protein. Along with that, there could be other polymers as well, such as trehalose, which is a disaccharide, a non-reducing disaccharide of glucose. And they contain a tube for host penetration. We are going to talk about that. And we're going to talk about the detailed examples and what they do in a second. This is just an overview of that. The other one, which is the chytridiomycota, is the flagellated cells in at least one stage of life cycle. And see that actually gave rise to the idea that these zoosporic fungi are once considered as protists. And the cells do have the chitin and beta 1316 glucans, means that these glucose molecules are joined together by 13 beta 13 or beta 16 linkages, and glycogen used as a storage of the carbohydrate. And sexual reproduction often results in a zygote that becomes a resting spore or a sporangium, and they could be saprophytic or parasitic. See right here, the microsporidia, we saw that they are obligate intracellular parasite. That means they have to be inside a cell to live. They are not free living. On the other hand, the chytridiomycota can be saprophytic. That means that they devour on the dead animals or plants. So they can be free living. And these are known as the decomposers. They live on dead material, or they can be parasitic as well. And we are going to talk about several one of them. After that, the next group that we are going to talk about is the zygomycetes or the zygomycetes fungus. So the zygomycetes, there are two groups that we talked about already. The mucormycota. So the thalli, usually filamentous and non-septate. So what is thalli? So thallus is single, thalli is the plural, and thallus is the fungal body. So that is a generalized term of the fungal body, and usually filamentous and non-septate. And this thallus, this non-septate filamentous, these structures are also known as the hypha singular, hyphae plural. So here we can just restructure this sentence this way, that the mucormycota thalli is hyphal, or mucormycota thalli usually is usually consists of non-septate hyphae. So this is the way we use these two terms, and we are going to talk about it back again. And sexual reproduction gives a thick-walled zygospore that are often ornamented. That means they have spikes on them, and we are going to talk about those when we talk about the zygomycetes in detail. So the other group of the zygomycetes is the glomeromycotina, and these are also filamentous. Most have these arbuscular endomycorrhizae. As I said, that arbuscular endomycorrhizae is essentially the endosymbionts of the vascular plants, and they help the plants to absorb nutrients, particularly nitrogen, and essential for their well-being. So they form exospore outside the host, and they lack centriole, conidia, or aerial spores. That means they do not have specific structures or aerial spores which spread the spores, because these are underneath the soil, essentially. So they are just spread through the soil. There is no aerial spores that can be that will be exposed to the air, because there is no possibility, as they are deep inside the soil. So the next group is known as the Dicaria, and we have two groups, Ascomycota or Ascomycetes. This sexual reproduction involves a meiosis of a diploid nucleus in an ascus, and this is essentially where these spores are. So we are going to see that these sac-like structures, and this give rise to the haploid ascospores, and most undergo the asexual reproduction with the formation of these conidiospores, which are essentially, we are going to have many different terms, and these are specialized aerial hyphae called conidiophores. So these on conidiophores, the conidiospores are essentially formed. And many produce with these ascospores within the complex fruiting bodies, which does have a sac, and these known as ascocarps. So you can have conidiospores, we can have ascospores, just depends upon the morphology of the particular fungi. This can be saprophytic, living on dead plant or animal bodies, and parasitic forms, and many form mutualistic relationship with the phototrophic microbes and form lichens. So these are the group which is Ascomycota. And some of the notable of them is this aspergillus, which can cause serious human ailment as well. Candida is also called many different fungal diseases or mycosis. On the other hand, particularly our species Neurospora crassa is one of the very standard laboratory experimental eukaryote that many experiments are done, or research is done with that, because it's a model standard organism. Also, just remember that many of the antibiotics are produced. Penicillium notatum is also one of these Ascomycota. And along with that, the Saccharomyces, there are many different. Saccharomyces cerevisiae is the baker's yeast, and essentially many of these are essential for the baking industries, for many of the fermentation industries. Many fermented foods use these fungus to ferment the foods. So essentially, very economically important group is Ascomycota is a very important, economically important group. So after that, we are looking at this Basidiomycota or the Basidiomycetes, and these are many common mushrooms and the shelf fungi. The sexual reproduction involves the formation of a basidium, which is a small club-shaped structure, typically forms spores at the end of tiny projections, and in which these haploid basidiospores are formed. Usually 4 spores per basidium, sometimes could be 1, 2, even 8, even more than that as well. And the sexual reproduction involves the fusion of opposing mating type and in a dikaryotic mycelium. That means that, as I said, because Ascomycota and Basidiomycota, these are the dikarya. So after the fusion of the two mycelium, or the two cells, essentially, the cell, a single cell, remain with 2 individual haploid nucleus for some time. So this is what it means, that the parental nuclei are paired but not initially fused, but essentially they are fused after some time, and there is a meiosis or reduction division by which these basidiospores are formed, which are haploid and can be dispersed by the form of asexual reproduction. So asexual reproduction and sexual reproduction goes in cycles depending upon the condition of the organism that we are going to discuss. This includes many plant pathogens which cause the rust and smuts belonging to this iridinomycetes and this ustelogenomycetes, and we are going to talk about what kind of disease they form in the subsequent part. So after that, we are going to look at the generalized idea about the fungal biology and their diversity. And the first important thing is that the fungi, they are terrestrial organisms with global distribution. And it is a very, very large group with 2 to 5 million different species. And every year also, even 20 new fungal pathogens for the human are actually described. So you can see that there is a huge, large group, and there are almost 5,000 plus pathogenic fungi which can infect the human beings, animals, and cause infection to the plants and the crops, and cause economic damage. So they are essentially saprophytes. Many of them are saprophytes, which secure nutrient from dead organic material by releasing the degradative enzymes. So these could be dead animals or dead plant material. And essentially, these are beneficial for us because what happens, just say, for example, from in a lawn, there are dead grasses underneath these green grasses. And these fungi, which are present in the soil, actually decompose those dead grasses. And this is a natural way to recycle many of these nutrients in the soil, and they are beneficial in that sense. They show a characteristic which is known as osmotrophy. So osmotrophy essentially means to push, but essentially what they do, they absorb the soluble products with their entire surface. So that's basically known as the osmotrophy. So they decompose the organic material to the simple organic compounds. So you can see that they can recycle the carbon, nitrogen, phosphorus, other critical constituents, and release and made available for the living organisms. And as I said, many are pathogenic in the animals. Some are beneficial relationship. Say, for example, the mycorrhizae we talked about, association with the plant roots. These are essential for the nitrogen fixation for many of the plants as well. So one of the important consideration for these is that they are formed in many different forms. So some of them could be mycelial, but some of them do have the form which is known as the yeast form or a single cellular form. So yeast form is a single cellular form of these, and the mycelial form, where the thalli or the thallus grows into filamentous structure, is the mycelial form. So these are the two nomenclatures. So industrially, they are actually essentially very important. So you can see that the fermentation yeast in making bread, wine, beer, cheese, soy sauce, many other things which is not named, organic acid like citric acid, gallic acid are produced with that. Certain drugs like an ergometrin. Ergometrin is used in obstetrics to reduce bleeding after childbirth. Cortisone. So these are some of the steroids that topical steroids. These are produced from the fungi. Then, of course, the penicillin was first discovered from Penicillium in Alexander Fleming, which essentially changed. The first antibiotic was discovered from the fungi, and many other fungi produced, like aggressiofulvin is another antibiotic which is produced, and there are many others. So essentially, the discovery of the antibiotics essentially changed the whole face how our biomedical science actually evolved over the period of time for the last 100 years. Some of the immunosuppressive drugs like cyclosporin, that is also produced by the fungi. So there are industrially very, very important. And along with that, they are very important research tools. Cytologists, geneticists, biochemists, biophysicists, and microbiologists use fungi in their research. And there are some notable forms of these fungus. So for example, the baker's yeast, Saccharomyces cerevisiae, and a couple of other species. But essentially, Saccharomyces, the yeast, is a very important study tool, and particularly almost all the discoveries related to the eukaryotic cell cycle and the control of the cell cycle were all done basically with the study in the yeast, because it is easy to grow in the laboratory, and they do have this eukaryotic cell, so it is easy to control, manage, and do the research using those. Neurospora crassa is another organism that is used also as research tools. So immensely important in industrial and research use. So after that, we are looking at these fungal structures. And as I said, the thallus is the name of the body or vegetative structure of the fungus. And the thallus and the thalli is plural. So how the thallus can be? The thallus can be in the yeast form, which is a single cell fungi, usually larger than bacteria because it's an eukaryotic cell. So yeast is a single cell. And again, yeast could be many different species of different fungi. But if they are in the single cellular form, we call them, they are in the yeast form. Whereas if the thalli or the thallus or the thalli appear to be in the mold form, these are the multicellular masses. And these have the hyphae that form the mycelium. So thallus, hypha is singular, hyphae is plural. So think about that. If this is a single hypha, this is right here is a single hypha, this is another hypha, then another hypha. When we are talking about them together, we call them as a hyphae. And the mass of the hyphae is known as a mycelium. So many different terms, but they are overlapping. So thallus contains the hyphae which form this mycelium. But the thallus could be either yeast or mold, which is essentially the hyphae and the mycelium. So just use these terms with the proper meaning of that. And sometimes there are too many terms which are overlapping and sometimes confuses us, but it is pretty simple. So thallus is essentially the body, depending upon that whether they are multicellular or unicellular, and the multicellular form has these long filamentous structures known as a hyphae, and the collection of the hyphae form the mycelium. So these mycelium can be aseptate. Right here, you can see that these mycelium does have the nucleus within them, and they do not have any septa or separation. Whereas here you can see that this is a hypha which is septate. So essentially, even if it's a septate hyphae, they do have these septa with a central channel through which the cytoplasms can move from one chamber to the other. On the other hand, here you can see that the septate hyphae also may have a disc-like structure with multiple pores in them, through which there is a continuity of the cytoplasm. So these are the overview of these structures. So after that, we are looking at these fungal hyphae, which also specifically tells us that there is an cenocytic or aseptate hyphae. So cenocytic or aseptate hyphae, this is right here. Protoplasm streams lengthwise through the hyphae uninterrupted by the cross walls. Whereas the septate hyphae have the cross wall called the septa. Single or multiple pores enable the cytoplasmic streaming. So after that, we are looking at the fungal reproduction. So the asexual reproduction as well as sexual reproduction, there are two types of reproductions that we observe in all the fungi. The asexual reproduction, the parent cell undergoes just simple mitosis and divides into the daughter cell. The mitosis in vegetative cell may be concurrent with budding to produce a daughter cell and may proceed through a spore form. So here you are seeing that transverse fissure forming a new cell wall right here. So let's take a look at what are the different types of asexual spores that are formed. So right here, when we are looking at this arthrochonidia or the arthrospores, here there are two septate hyphae, and these septate hyphae just divide by itself, and the transverse fissure forming a new cell wall. And these individual structures, when dispersed right here, these are known as the arthrochonidia or arthrospores. So this is the vegetative body, and when the vegetative body's individual septate hyphae actually disperse by themselves, they are known as the arthroconidia. The entire structure, which is the cellular structure along with the nucleus, when the entire structure is known as the arthroconidia, which is also interchangeably used as the word arthrospores. Whereas another type of asexual spores that is formed in many fungi. Here you are seeing that at the end of the mycelium, so here you can see that this is the extension of the mycelium, a fruiting body-like structure is formed, and this is known as a sporangium. So this is a sac-like structure inside which these sporangiospores, which does have haploid or a single copy of the nucleus, or the single copy of the DNA in the nucleus. These haploid spores are formed. When mature, this sporangium essentially bursts and the sporangiospores are dispersed. So this is the sporangium, which is a sac-like structure. On the other hand, the conidiospores, these are also similar to sporangiospores, but they are not enclosed in a sac, but produced on the side of the hyphae. So here you can see that this is essentially the mycelium, and one of the hyphae is essentially extending into the structure, and each of these spores are attached to each other and grow in chains, essentially. And these are known as the conidiospores. And the structure which essentially support these conidiospores in this finger-like projection, this structure is known as a conidiophore. So conidiophore is basically the stalk upon which the conidiospores basically germinate and dispersed. So on the other hand, the same way, the stalk of the sporangium is known as a sporangiophore. So if you see the word phore, that means that is the stalk. The sporangium is the sac, sporangiospores are the individual spores, where conidiophore is the stalk, and the conidiospores develop on that stalk. On the other hand, the blastospores produce from a vegetative mother cell by budding. So here you can see that this is a septate hyphae, vegetative mother cell, and from here, on both sides of each of these hypha, this is budding, and on the top of the bud, these blastospores are essentially formed. Sometimes these are also known as blastoconidia, because these are also not enclosed in a sac, and they are naked spores. So just remember that the conidiospores and the blastospores are naked spores, whereas the sporangiospores are essentially within a sac or a sporangium. That should be more than enough for the different classification of the asexual spores. In contrast to the asexual reproduction of the fungi, the sexual reproduction involves the fusion of the compatible nuclei. And what we mean by the compatible nuclei? There could be two different types of sexual reproduction that is observed in the fungi. One is a homothallic fusion. So in the homothallic sexual reproduction, form sexually compatible gametes on the same mycelium. That means that you have the same fungus, and one part where you have one nucleus, another part you have another nucleus, and they actually come together. And this is essentially self-fertilization. So if it is the same fungus, or the homo means the same body of the fungus, essentially mate and form these the union of the two gametes, or the two nucleus happens. We call this as a homothallic sexual reproduction. Whereas the heterothallic sexual reproduction requires the outcrossing between two different yet compatible mycelia. So these are the two different mycelia. It is the same species that are growing next to each other, but the thallus, or the body, is different. So for an example, we have the same species of a mucor. One is considered as an A plus because we do not have a male or female. Another next body going to it, we can just name it as a A minus. So when A plus mate with A minus, we call this as a heterothallic sexual reproduction. So the sexual fusion may occur between the haploid gametes, which is a single copy of the DNA, and they form this gametangia, or the gamete producing cells, or the hyphae. Interestingly, what happens that in some stages of these fungal fusion, the two nuclei do not fuse immediately, but it takes some time to form these gametangia or gamete producing cells. So we will mean what? What do we mean by that? So let's take a look at this structure. So suppose a fungal structure does have the haploid stage or the N. And the haploid stage, the N, which is the single copy of the DNA that is present in the nucleus, this reproduces by asexual reproduction. That means that they produce all the different ascospores, they produce the conidiospores, they produce the sporangiospores, and they produce blastospores. So all the different asexual reproduction that we essentially saw in here, that can happen with this haploid stage. And how it happens, because this is a simple mitosis, which is the equal division, and each mitosis produces this n number of nucleus, which forms into a spores and spread. Now, in the sexual reproduction, there is a cytoplasmic fusion, either homothallic or heterothallic. At this stage, one nucleus N meet with another nucleus N, and there is a dikaryotic stage. What it means that the two nucleus, unlike what happens in an animal reproduction, the two gametes actually fuse to form a zygote immediately. There is a stage which is dikaryotic stage, where the two nuclei actually exist for a certain period of time. So the zygote does not form immediately. After some time, there is a nuclear, so cytoplasmic fusion happen, but nuclear fusion doesn't happen immediately. Once the nuclear fusion happens, it forms the zygote, which is the diploid stage. So this diploid stage is the result of sexual reproduction. Once the diploid stage is reached, this will be a reduction division or meiosis, which will again form these haploid spores. And essentially, there are 1 to 4 individual spores formed from one nucleus, but there are meiosis followed by mitosis, which can form even 8 to 12 spores out of one nucleus as well. So this is essentially the life cycle or the sexual reproduction, which cycles will along with the asexual reproduction of the fungi. And the sexual and the asexual reproduction essentially is observed in depending upon that what kind of environment the fungi is. Depending upon when their nutrients are abundant, usually, and there is an optimum temperature and everything is good, usually the fungi reproduce by this haploid stage forming the spores. Whereas when there is the nutrient shortage or the environment is not essentially favorable at that time, sometimes there is a sexual reproduction that is observed. But that is not universal. Sometimes it is seen, sometimes the sexual reproduction and the asexual reproduction essentially cycle without these requirements as well. So after that, we are looking at a certain group, which is the zoosporic fungi, and these produce these motile spores. So zoosporic essentially mean that they do have at certain time during the evolutionary process does have some flagella, and they were considered as protist. And protist, which is a protozoa animal, and the zoo essentially is due to these motile spores, they were termed as the zoospores essentially. So one of the group of this zoosporic fungi is the microsporidia, and again they reside in they are endoparasitic fungi, and they must infect the host cell to complete their life cycle. And here you are looking at a typical structure of this. Here, this is a anchoring disc, which is connected to a long structure, which is known as a polar tube or the polar filament. So this polar filament essentially burst out of this spore and essentially drive ejected with a force and pierce the host cell membrane and allow the parasite to go inside. So here you can see that there are some structures like this. You have these exosporic structure, endosporic structure. You have a nucleus. They do not have the other organelle like a mitochondria, centriole. Those type of organelles are not present in there. And there is a vesicular, these structures are known as a vesicular polaroplast, and these are known as this lamellar polaroplast, and there are usually a posterior vacuole is usually seen. And these spores can be rod shaped, they could be a little bit filamentous, they could be round, and they could be even egg shaped structures as well. And the spore germination essentially happens only in the host. As all the microsporidia are endoparasitic fungi, they have a very reduced genome size and the complexity. Another group which is also important, which is within the zoosporic fungi, which is Rosellomycota. We are not talking about the Rosellomycota in here, but that is also another group of fungi, which is also zoosporic fungi. And we are mainly going to talk about this microsporidia and the other group that we have, which is the Chytridiomycota. Chytridiomycota burst into the news lately. The reason for that, they are affecting the biodiversity. And we are going to explain that what we mean by this. They're free-living, saprophytic, and parasitic forms infect aquatic plants, animal, insects. Sometimes they're found in the anoxic rumen of the herbivores, and they contain a single posterior whiplash flagellum, and that's one of the reasons that they are zoosporic. They do have the sexual asexual reproduction, and this is the most important thing, that the amphibians succumb to the chytrid infection. And this is lately bursting into the news of these Chytridiomycetes. Along with that, there could be other type of, there are several other Chytridiomycetes which are of importance. One of them is also known as this enterocytozoon and BNC. And this enterocytozoon, as you can see, that if they are ingested, they can cause diarrhea. If they are inhaled, they can cause the pneumonia as well. There is also another group which is known as another genus known as the encephalocytozoon, and this encephalocytozoon, as the name suggests, that can cause encephalitis as well as nephritis. And these are the source of these Chytridiomycetes are the aquatic birds. So usually a flock of aquatic birds, when they visit a water body, they actually release a large number of these Chytridiomycetes in this water bodies. So another important thing that what we were talking about, that what is going on with the amphibians. So lately, what has been seen that many of these tropical rainforests and many in different continents across the globe, the amphibians are attacked by these Chytridiomycetes. What the Chytridiomycetes essentially does, that they actually cause a skin problem, which actually the skin of these amphibians essentially get thickened or get hyperkeratinized. And essentially what happens, that impedes their breathing. What it means that the amphibians actually do gaseous exchange with their thin dermis, which is the skin. So skin is essential for them for the gaseous exchange. And when these the dermis or the skin essentially become hyperkeratinized or thickened by this chytrid infection, that impedes the gaseous exchange. So the blood electrolytes get imbalanced, and essentially due to that, they suffer from the cardiac arrest. And you can just look at the YouTube and some of the newses. You will see that this is essentially threatening the biodiversity across the world, and many species may essentially be extinct within our lifetime just due to this chytrid infection. And why the chytrids essentially chose these, specifically the amphibians, that is an open question. That is pretty much all about the zoosporic fungi. Next, we are going to talk about the zygomycetes fungi. And these does have a synocytic or accepted hyphae. And we're going to talk about what their distributions are. Rhizopus life cycle, which is a typical representative of the zygomycetes, and benefit, harms, functional importance of these glomeromycotina, which are the mycorrhizae, and helps the plants to absorb nutrients. And also, we're going to talk about these plant fungus symbiosis. So they do have these alternative mechanism of spore dispersal, which is either by wind or they can cling to the animal. And the sexual reproduction actually occurs when the environmental conditions are not favorable, whereas these forms these zygospores, which remain dormant in unfavorable conditions. And these zygospores are essentially thick-walled structures, sometimes decorated, and we're going to talk about that how they are formed. And the hyphae actually lack the septums or the divisions, and that's why they are called the synocytic or accepted hyphae. And we're going to talk about the two groups, which are the mucormycota and glomeromycotina. There is also another group which infects many of the insects, animals, which is also worth mentioning, which is called the zoopagomycota, but we are not going to talk about that essentially. So let's take a look at the mucormycota, and these include many of the fungi of industrial importance, and they are both free-living saprophytes. That means they live on the dead plants and animals, and can be parasitic as well. Food industry can be used, but also cause the food spoilage. So they're all over the place. And the ectomycorrhizae, which form this mutualistic relationship with the plants, are also members of this group. Now, the mycorrhizae is a general term, which is the fungus associated with the plants. And it could be two different types. One is an ectomycorrhizae, which is usually their hyphae is outside the plant. And it could be also the endomycorrhizae, which is also found in many of the vascular plants, which is inside the root system. So endo and ectomycorrhizae, and the ectomycorrhizae is also belong to this group. So the Rhizopus, say for example, this Rhizopus stolonifer essentially is a classic mucor or the mucormycota or mucormycetes, and these grows on the substrate of moist carbohydrate rich food like bread. So when you see this blackish kind of a bread mold, usually those are the Rhizopus, not this blue-green mold. That is usually something else, which is the Ascomycetes, which is belong to mainly the penicillium. We're going to cover that later. But these are the black molds which can happen on the bread surface. So the hyphae actually quickly cover the surface, and the rhizoids, or the portion of the hyphae which absorb the nutrients, are called the rhizoids. They essentially absorb the nutrients from this carbohydrate-rich substrate, and essentially the stolon is essentially the erect hyphae, and the hyphae which essentially goes horizontally are essentially form the rhizoid structure. So here we are looking at these. So let's point out. So here what we are looking at, that this is a typical Rhizopus. Many of the mucor species also look like this. So here what you are seeing, this erect part, that this is essentially the stolon, the erect part, whereas right here, which are these are the rhizoids which penetrate the substratum and absorb the nutrients. The sporangium during the asexual reproduction forms at the tip of these stolons, and you can see that these sporangiums are on the sporangiophores, and these sporangium on the sac, when they burst, the asexual spores essentially come out. When they fall onto the substrate, they will essentially germinate. So this asexual reproduction is the norm when there is the favorable condition and the nutrients are available. But if there is a condition when there is a lack of nutrients and the environmental condition is not favorable, at that time also the sexual reproduction can follow. So here what we are looking at, there are two separate individual of the same species and say this is a plus mating strain, and there is a minus mating strain, plus and minus. And due to the pheromone secretion, these are secreted, chemicals secreted by these individuals, pheromone secretion, the two hyphae come together, and the two individual hypha, they come together and they meet, and this essentially is known as the plasmogamy, or the union of the cytoplasm. So once the plasmogamy essentially happens, that there is a structure which is formed, which is this gametangia. This gametangia is an elaborate structures with a thick cell wall. Sometimes they are spiky and large, and they are much more stable compared to these sporangiospores in nature. So they can actually withstand adverse condition in nature. So essentially, these individual nucleus, which are the haploid, essentially remain for some time in this dikaryotic stage without unfused. And then finally, when before germination, there is a process which is the karyogamy happens, first plasmogamy, and then after the fertilization, there is a dikaryotic stage. After the karyogamy, the two nucleus fuses to form a diploid nucleus, which undergoes a meiosis, followed by, of course, the mitosis, which form essentially from these diploid nucleus four individual spores from one nucleus. And there is a germinating zygosporangium that is formed from this zygospore, and these spores essentially disperse and they will germinate into new vegetative bodies, or the thallus of this rhizopus, mucor, and this is a typical life cycle of the zygomycetes. So there are, in the mucormycota, there are different types of fungi which are important. Say, for example, this Rhizopus chinensis cause this seedling blight rice disease. But essentially, there is a bacterium, a proteobacterium, which is Burkholderia, that essentially is present in this Rhizopus and produces this toxin. So other than that, this Rhizopus oligosporus is used to produce this tempeh. This tempeh is essentially a fermented form of the soybean, which is found in Indonesia. Indonesia, this is also a food, and also used with soybeans to make these sufu curd. So this is essentially a fermented bean curd. So for an example, the tofu, which is essentially the cheese-like structure, or the cheese-like food, which is essentially made by the precipitation of the proteins from the soybean proteinaceous part. So these tofus, these can be fermented, and many of these rhizopus, including this sufu curd, these are the bean curds, but they are fermented. These are formed by the Rhizopus oligosporus can also make them. Along with that, commercially, they can produce different kind of anesthetics, birth control. They are source of all different types of alcohols. They can produce meat tenderizers because they are saprophytic and produce these enzymes. And yellow coloring in the butter or the margarine, which is the artificial butter. These are some of the other use or the commercial importance of this group, which is the mucormycota. So after that, we are going to talk about another group, which is the Glomeromycotina. These are the mycorrhizal symbionts and of vascular plants. These are usually herbaceous plants and tropical trees. So the mycorrhizal fungi usually grow in association with the roots and their plant's holes, and this is a mutualistic relationship. So essentially, the fungus delivers the nutrient to the plants, and the plant provides the carbohydrates to the fungus. And these essentially, the Glomeromycotina is primary, the reproduction is asexual reproduction. The sexual reproduction has not been observed. And there are some specific type of structures, which is known as the apressoria. And we are also going to see these kind of structures when we talk about these basidiomycetes later part. These are specialized flat type of hyphae, and these essentially penetrate their host plants. And in this context, this will be the root system. And this symbiotic relationship is almost essential for many of these plants for their normal survival. So after that is we are going to take a look at the most diverse group, which is essentially the dikarya. So that we are going to talk about its ecological, economic, medical importance. Saccharomyces cerevisiae, which is the baker's yeast. We're going to take a look at their life cycle. And we are going to talk about this Ascomycetes. That's one of the group out of those. And we are just going to find the word sclerotia, define or understand that what is the St. Anthony's fire, LSD, sick building syndrome. These are all the byproducts from these fungi. And we are also going to talk about this Basidiomycetes, which is another group. So Ascomycetes and Basidiomycetes, we're going to talk about that and identify some of the fungi which cause plants rust and the smut and the rust disease. So let's take a look at these in a little bit detail. So as I said, that this dikarya does have a dikaryotic stage, but we have also seen this group which with overlapping. So in the previous group, which is essentially the Rhizopa species, we also do see that there is a stage which is the dikaryotic stage. We also see that, but these are grouped in a different way phylogenetically, and the zygomycetes do not fall under the dikarya. So the cell membrane does have the sterol, and this is ergosterol. So this is very different that for mainly the animal plasma membrane that what we have, that we have the phospholipid. Along with that, we have cholesterol. On the other hand, these cell membrane of this dikarya contain the ergosterol. So essentially, targeting this ergosterol synthesizing machinery essentially are found in many of the antifungal creams or antifungal medications. And these actually work very well, particularly for the human pathogen. And these are important drug target which can cause disease, because ergosterol is not found in the human or the animal tissues. These are very specific in the eukaryotic, but specific to the fungi. And the reproduction methods alternate between the haploid and the diploid. And we have talked about this diploid and haploid life cycle, and we are going to go to the specifics of them in a second. And there are two important taxa that we are going to talk about, is the Ascomycota or the Ascomycetes, and the Basidiomycota or the Basidiomycetes. So the Ascomycota is essentially, the ascus is basically a sac. So this is basically a sac fungi, and they are found in freshwater, marine, and terrestrial habitats. And their characteristic reproductive structure, which contain the ascus, which contain these ascospores. And they could be very brightly colored, sometimes red, brown, blue, green molds cause the food spoilage, and sometimes they are very, very commercially important. And there are also just fungi that infect most the most human pathogens belong to this Ascomycetes as well. So let's take a look at this Ascomycetes life cycle. So this is a typical Saccharomyces cerevisiae, which is the baker yeast, and these baker's yeast belong to this Ascomycetes group, and it could be in the single cell form, which is the yeast, whereas in unfavorable condition, it can also become filamentous, which is with the mycelium, and the mycelium are usually septate in these cases. So the nutrient-rich condition, the mitosis and budding occurs at unscarred region of these yeast cells. And once the entire yeast cell, see that here you have multiple scars, when the entire surface of the yeast cell actually are scarred, there will be no more budding, and the senescence sets in, and that yeast cell will essentially die. On the other hand, in the nutrient-poor region, the meiosis happens and the haploid ascus containing these ascospores are formed, and this produce these haploid cells which remain bound within a common cell wall, which is called the ascus. So let's take a look at this life cycle. So here what we are looking at, this is an ascus containing 4 haploid ascospores, and these arise from 2 different parents. That's why they are AA and they are alpha. So suppose alpha mating type ascospore is released, it germinates, and this is the vegetative life cycle. So in the vegetative life cycle, which is the haploid life cycle, and in this condition, what can happen that one yeast buds into the other. As we can see that this is right here is a budding of the yeast. There is another budding of the yeast, and sometimes this is another budding, and sometimes they remain attached to each other and form long chains before they are separated out. But eventually they will separate out. And this is the vegetative life cycle that will go on. On the other hand, this is an A mating type, contrast to the alpha, and the same way the vegetative life cycle will go on. But in the nutrient poor condition, what happens that this alpha and the A, they actually form the zygote. And this is the diploid life cycle. Now, this diploid life cycle, sometimes, as I said, they can be in the yeast form, or sometimes they can form the mycelial form, or they can just become a mycelium because that is more favorable for their survival. And this is essentially the diploid life cycle. And from the diploid life cycle, if there is a nutrient-rich condition returns, this dikaryotic stage or the diploid stage, they will undergo the meiosis, and from the diploid DNA, they will form 4 haploid spores which are within the ascus, and they will germinate to form the vegetative haploid life cycle. So this is essentially the ascomycetes or ascomycota life cycle. So there are several different kinds of structure that is formed, and this is the filamentous form of the life cycle that I was talking about. And they form this conidia or the asexual spores, and the sexual reproduction happens when the opposite mating types actually meet. And the hyphae from one type is known as an ascogonium, and the hyphae from the other type known as antheridium, and these essentially meet together. So the ascogenous hyphae is fertilized the ascogonium hyphae essentially. And then it forms into an ascocarp, which does have a cup-like structure, and essentially that disperses. Sclerotia is, on the other hand, is the masses of hyphae which can survive the stressful condition. As I said, that many of these ascomycetes can live in the yeast form, but they can be dimorphic. That means not dikaryotic, dimorphic. That means they can change their morphology from a single cell yeast to the sclerotia, which is the masses of hyphae to survive the stressful condition. And the classic example of many of these are, say, for example, the penicillium is one of the example of these ascomycetes, and we're going to see a couple of different forms as well. So here we are looking at these ascomycetes. So let's start with the asexual reproduction. So here you are looking at the hyphae and see that these are the septate hyphae. And the septate hyphae actually form this structure, which is the conidia, which are the naked spores. And these naked spores contain the haploid DNA. And when they essentially are received, or they essentially fall on a substrate, they will germinate and they form the same hyphae. So here is one individual of the same species, and this is another individual of the same species. This is a minus mating strain, this is a plus mating strain. And in a certain condition, they form, particularly when the nutrient deprived condition, they will secrete some of the pheromone, and these structures will be formed from these hyphae, which is an antheridium and the ascogonium. and between the plus and the minus mating strains. Now, the fertilization actually occurs, which is the plasmogamy. And this is the dikaryotic hyphae, and this, which is essentially from the ascogonium. So these dikaryotic hyphae, the spores essentially do not fuse immediately, but gradually this structure develop from here into an ascocarp, which contain this dikaryotic or ascogenic, say for an example like here, hyphae, and some of them are sterile hyphae that do not contain this dikarya. So this ascocarp contain both. This is inside this cup-like structure. And now you can see that this is an young ascus, which is still in a dikaryotic stage. The two nucleus has not fused yet. So finally, at this stage, the karyogamy happens, which is the two nucleus actually form the zygote. Once that happens, it undergoes the meiosis. So the first meiotic division from the 2N, it becomes N. Then there is one mitotic division, which is we call as a meiosis 2 or the mitotic division 1 after the meiosis or the reduction division. So essentially, 4 haploid spores are formed. So just make sure that I'm clear, just quick basic cell biology, that from the 2N diploid, the first meiotic division produces 2 nucleus with N. These 2 nucleus, each one of them undergo a round of mitosis, which is equal division, which is the part of the meiosis known as the meiosis 2. The meiosis 1 is the reduction division from 2N to N. Meiosis 2 is the mitotic division, which is from N, it produces another N. So from initial 2N, it produces 4N or 4 haploid nucleus. So essentially, once that is divided, each one of them will form an ascospore. They will essentially released and they will germinate into these filamentous ascomycetes, which can reproduce by asexual reproduction as well and go into the sexual reproduction as the according to the environmental condition. So the genus Aspergillus is very, very common, and this is a very, very common, widely known ascomycetes, and this is environmentally ubiquitous, and these are human pathogens, cause allergy, asthma, sinusitis. Sometimes in immunocompromised patients, this could be really fatal as well. On the other hand, another Aspergillus nidulans is a model organism to study eukaryotic cell and developmental biology that is used. On the other hand, there are some other species like Aspergillus oryzae. This produces fermented food. Say, for an example, from the rice, by fermentation, it causes the Japanese drink, which is the sake, and also production of the soy sauce, as well as the miso, which is essentially a base of this miso soup, Japanese in the Japanese culture, in the Japanese food. These are essentially fermented soybean products from these, which is done by these Aspergillus species as well. So they are also important in biotechnology, and because they can be genetically manipulated and can produce industrially important products. So there are more pathogenic ascomycetes, and one of them is the Claviceps purpurea. And these parasites essentially forms on rye and other grasses. And this formed this ergot, which is the toxic condition from eating this infected grain known as the ergotism. And this ergotism is essentially known as the St. Anthony's fire and killed thousands of people during the Middle Ages in Europe. So these are extremely potent and lethal toxins are produced by these. Also, there are pharmacological activities are due to the active ingredient, which is known as this lysergic acid diethylamide, LSD, common street name. This is one of the recreational drugs that is being used, which is known as acid, and this, which is at a certain amount, can cause hallucination as well and an euphoric experience. So essentially, these are found in some of these fungus as well. Also, along with that, the worth mentioning are the candida. For the example, the candida albicans that can cause a lot of different type of skin infection, including the ringworm. This is the candidiasis, is a very common type, which is called the jock itch, the athlete's foot ringworm. This is caused by the candida. Then blastomyces and the histoplasma. The histoplasma can cause serious type of subdermal infection, and some of these can cause also the thrush that is known. These are all human pathogen. Along with that, there are some of the extremely potent pathogens. Stachybotrys is one of them. The Stachybotrys is found in wood panels in houses, and essentially it produces these spores, which essentially do not germinate and that stay put. But what happens that sometimes if they are exposed to water bodies, they essentially they will germinate and they will spread, and this cause a sick building syndrome. And I have put forensic files, which will be for your entertainment, in the supplementary material, and you can just take a look at that what these Stachybotrys can do to a building. And Aspergillus, some of the species also produce these aflatoxins, and these aflatoxins are extremely potent toxin and cause liver toxicity, and they are cancer causing chemicals, that they are carcinogen. And also the Fusarium is another species which are called the fumosins, and all of these are extremely potent fungal toxin. And in general, these fungal toxins are known as the mycotoxins. Very important as well. So these are some of the consideration of these group, which is the Ascomycota. Also, along with that, this is important for the bats of the Northern America, and these are the part of our ecology. And sometimes in the evenings of the summertime, these small bats, which are usually in different caves, they just come out. And there is a white fungal hyphae actually grown around these bat muzzles. And this is caused by this Pseudogymnosascus destructans. And they erode the epidermis before they invade the skin and the connective tissue. Essentially, they will kill the bats. And essentially here you can see a part of the bat wing is invaded by this fungus, and essentially that may eliminate these bat population, the native bat population, by 2026. So these some of these funguses, as we have seen earlier, particularly with the chytrids as well, that they actually threaten our biodiversity as well. So very important. And the reason is that many times this is attributed to the human encroachment. The bats actually do live in some of the caves and the mines. Sometimes human just go and visit in there, and along with the temperature rise globally, sometimes these bats find another places from one to another, and some of these infected bat can bring these fungi from one place to the other. So the displaced bat can actually cause the spread of the disease as well. The next group and the last group is the Basidiomycota and the Basidiomycetes, or the club fungi. And these include the jelly fungi, the rust, the shelf fungi, stinkhorns, puffball, toadstools, mushrooms, bird's nest, a wide variety of macroscopic fungi, which can be very large as well. So the basidium is essentially the structure at the tip of the hyphae, which is essentially the club-shaped structures, which form these basidiospores, millions of these basidiospores. And the basidiocarps are the fruiting body which hold the basidia essentially. So here what we are looking at, that how it is formed. This is a typical mushroom that is fully formed. This is a basidiocarp. So below below the structure or the cap of the mushroom, you can see that these are the gills, and these are lined by the basidia. And these basidia is essentially this club-like structure. This is the club-like structure that we are talking about. And it essentially contains the two individual nucleus, and they essentially undergo the karyogamy and form the zygote. This is a 2n diploid nucleus. This will undergo the meiosis, and you can essentially see that the basidiospores will be formed from there, which is the haploid. So this is the basidium, and this is the stigma, and these are the basidiospores, which will be dispersed. So essentially, once these mushroom essentially come out, within a short period of time, it will undergo this life cycle. And this is the stage at which millions of these spores or the basidiospores will be dispersed, and they will essentially fall onto the substrate, and the germination will happen. And these are the monokaryotic or N number. The nucleus is N number. And there are mating strains which can essentially meet with each other the same way. And the secondary mycelium, this will be the dikaryotic stage. So the dikaryotic stage will go on through the basidiocarp formation, where the nucleus essentially do not fuse. So in this part, even after mating, this is essentially the dikaryotic stage. Whereas after the basidium matures, the karyogamy happens, followed by the meiosis and the disbursement of the basidiospores. So this is the overview of the life cycle of the Basidiomycetes. So they definitely impact the human beings. Many of them are saprophytes. So when they are saprophytes, they essentially decompose the plant debris, especially the cellulose and the lignin. And these are essential for our environment. And these mushrooms essentially help our lawn to thrive properly as well. And many of these Basidiomycetes are the eatable mushrooms as well. And these are multi-million-dollar industry. You can see that there are so many different types of eatable mushrooms. But along with that, there are several non-eatable mushrooms as well. And these non-eatable mushrooms, on the other hand, can produce these mycotoxins like aflatoxins, and these are extremely poisonous and hallucinogen, and often these are lethal. Along with that, they can be pathogen of the humans as well as the plants, and the plant pathogens usually called these rusts and the smuts. So here, what you are seeing, that these can affect the corn, and instead of the corn producing the kernels, you can see that instead of kernels, they produce these large bodies or the large tumor-like structures, and these are essentially due to this Basidiomycetes infection. And these are the two group, which is the Iridinomycetes and then Ustilaginomycetes. These are the two groups which are called the rust and the smuts, and they are dimorphic in nature. So what we mean by that they can live, what we mean by dimorphic, that they can live in the yeast form, single cellular form right here, or they can be in the mycelial form. So the yeast must mate to produce the infectious filamentous dicaryons, and these essentially trigger the plant to form the tumors where the fungus produces these teleospores. So let's take a look at the life cycle of that. So we will start with this haploid sporidia, and this is the yeast or the single cellular form. So these single cellular yeast essentially form a conjugation tube, and they will go into a dikaryotic filamentous form, which is the mycelial form. The nucleus, although a plasmogamy happens here and division, but the nucleus remain as a dikarya, which is N plus N, two nucleus. And these structure essentially form, which is known as this aprosoria. We already talked about this when we talked about the mycorrhiza. Aprosorium, singular, aprosoria is plural. These are the structure from the mycelium which can penetrate the plant and go inside, deep inside, to essentially absorb the nutrients from deep inside the plant. And these aprosoria essentially penetrate the entire plant infectious part. And this, you can see that these are proliferation and branching, and these are the septate hyphae and form this septate mycelium. And from here, what happens that there is the union of the karyogamy happens as well, and this karyogamy produces the teleospores, which are essentially the diploid spores. And these diploid spores are dispersed from these hyphal structures. These are the teleospores, and these teleospores essentially forms the tumors in the plants. And essentially, these teleospores are dispersed from these structures, and these spores essentially germinate. And meiosis is followed by this, which essentially from the diploid stage forms the haploid sporidia, which essentially is the yeast form. And it must produce the infectious filamentous dicaryons to infect the plants. So this is the life cycle of the iridinomycetes and ustilaginomycetes, which essentially cause rust and smut in many plants. So essentially, what is the important part is that these are the fungi are that incredibly important to life on this planet, as they recycle nutrients, decomposition processes, and sometimes they can grow undesirably, causing disease. But essentially, they are also industrially important and produce many products that's essential for us. So here, this textbook gives you an overview of the main groups of fungi and how they operate, which can cause problems of human plants, and an overview of that. But the classification of fungi is extremely complicated, because there is no universal classification of the fungi. So I will just add a supplementary fungal classification paper, which came out in 2021 in the Nature Microbiology. And you will have an idea that even after sequencing, why making a classification of fungi is so difficult, because many of these groups of fungi overlap each other. So most of the mycologists essentially group these fungi according to the way they want to study, which are important, which do have similar type of structures. So these are artificial classification, because a real classification or evolutionary tree is very difficult to do with the fungal classification. So the taxonomy is incredibly complicated. And that's the reason that every 4 years, the International Mycologist Congress actually meet every 4 years. And in these large meetings, the taxonomy is discussed all the time, and it is being approved. And every 4 years, the latest taxonomic relationship in the fungus actually comes up. And depending upon how you study the mycology, different textbook will classify the fungus according to the approach of that textbook. So just to make sure that you are aware with that the classification or the way we presented the fungi in this textbook of the 12th edition of the Prescott's textbook, this may not be exactly the same. If you just consult a different textbook, they may classify the fungi in a little bit a different way. Basic understanding of the fungi and the characteristics are the same. It's just the grouping could be a little bit different.